So welcome, this is going to be our screencast for chapter 26, and in 26 we are going to be looking at the area of science called phylogeny. Now phylogeny is simply an area of science where we make comparisons between different species. And as we make those comparisons, what we often end up doing is we often end up creating something called the tree of life. As I had said, the definition for phylogeny is simply the evolutionary history of a species. And as we look at that evolutionary history, oftentimes instead of just looking at one species, we make comparisons between a group of related species. Now, in addition to phylogeny and making those comparisons, scientists often will take it a step further and they'll utilize the area of science called systematics. Now, the way systematics is a little bit different from phylogeny is because instead of just making simple comparisons between different organisms, they're also going to take those comparisons and hopefully determine their evolutionary relationship with each other. Now, the types of evidence that they'll use to actually demonstrate these different relationships would be fossil evidence, possible molecular evidence, maybe looking at different proteins, maybe looking at the similarities between amino acids between organisms, and then of course looking at the genetic data, so comparing DNA. And they use this information to infer or suggest these evolutionary relationships. Now, another area of science we need to be aware of is something called taxonomy. Now, sometimes people will get taxonomy and phylogeny confused, but taxonomy is simply the order, division, and actually the naming of these organisms. And Carolus Linnaeus is a really famous scientist who actually was one of the pioneers of taxonomy. And he had published a system of taxonomy that was simply based on resemblances between different organisms. Now, with the information that was gathered by Linnaeus, he also created a two-part scientific naming system. And this two-part system was simply to utilize something called the genus and the species for each organism. Now, the way that we utilize this naming system is that whenever you look at the scientific name of an organism, the genus itself will always have the first letter in that genus name capitalized, and the entire species name in that scientific name will be italicized. Now, both parts put together will be considered the name of that species, and this is also sometimes called the specific epithet of that species. Now, the type of classification scheme that was introduced by Linnaeus is considered a hierarchical type of classification. And this is a system for grouping species in increasingly broad categories. And that's what you're going to see over here on the right. So the taxonomic groups that he had actually created are going to go from something that's very broad, as we had said, to something very specific. So in this case, if you look down here towards the bottom, we have something called the domain. And the domain is going to be the broadest category that that particular species belongs to. Underneath domain, you're going to find the kingdom. Of course, underneath kingdom, you're going to find the phylum. Underneath the phylum, you're going to have class. Then, of course, comes order, the family, the genus, and then, of course, the most specific part of this classification scheme is going to be the species. Now, each taxonomic unit at any of the hierarchical levels in this classification scheme is considered a taxon for that scheme. Now, when you begin to link classification and phylogeny, again, this is going to help us to sort of infer these evolutionary relationships between these different organisms. Now, as we had said, systematics are going to depict these evolutionary relationships in a special type of diagram, and this diagram is going to be called a phylogenetic tree. Now, this is going to recognize only the groups that actually share a common ancestor, then, of course, all of the descendants from that group. Now, if you notice down here, we have a couple of examples of phylogenetic trees. Now, sometimes they consider this a rooted type of tree. And the reason they consider it a rooted tree is because it includes a branch to represent the last common ancestor of all the different taxa that are going to be found in the tree. Now, this phylogenetic tree is going to represent a hypothesis, as we said, about the evolutionary relationships between different organisms. Now, each branch point is going to represent the divergence of two species. And so if you look right here, this is going to be considered the most recent common ancestor for these two particular branches on this tree, for taxon B and taxon C. These are sometimes called sister taxa. And the reason we call them sister taxa is because they actually share an immediate common ancestor between the two, as we had said. But if you look over here, this is going to be considered a common ancestor between A, B, and C. And then, of course, if you look all the way back here off to the left, this is going to be the common ancestor for A through F. Now, if you notice, each branch point is given a special name, and we call that a node. Now, over here on the right, this phylogenetic tree just takes into account different species of animals, of course, and we look at the classification scheme that we had looked at in the previous screen, that hierarchical classification. We have species, we have genus, we have family, and we have order. And if you notice, the way this phylogenetic tree is set up, this is going to be the furthest common ancestor for all of the different species you see on the far right. And then if you were just to take these two individuals at the very bottom, this is going to be considered the most common ancestor for these two individuals. And again, as we had said, the way this one is set up, it's going to infer each of the different hierarchical categories that you see within this particular diagram. Now, there are some things that we can do with phylogenetic trees, and there are definitely some things that we cannot do. Phylogenetic trees definitely do show us a pattern of descent for the organisms that we are making comparisons between. But phylogenetic trees do not indicate when a species evolved. In other words, we can't associate a period of time to it, or how much genetic change has actually occurred within that lineage. So we need to make sure that we don't assume that a taxon evolved from the taxon next to it. So in other words, if you look down here towards the bottom, we have these different taxons that are located right next to each other, but again, we're not saying that this one necessarily evolved from this one. Phylogeny does provide important information about similar characteristics in closely related species, but again, it really doesn't make an inference in regards to time. Now, phylogenies are going to be inferred from various different types of data. So they're going to be inferred from morphological data and molecular data. Organisms with similar morphologies, in other words, body forms, or DNA sequences are likely to be more closely related than organisms with different structures, or again, DNA sequences. When constructing a phylogeny, systematics needs to distinguish whether the similarity is actually the result of homology, which we had looked at in the previous chapter, or analogy. Now, if you notice, we have these two words listed here. The definition for homology is simply due to a shared ancestry between the organisms. But the analogy that we see here is going to be a similarity due to something called convergent evolution. Now, down here towards the bottom, you see some diagrams that represent the difference between the two. When you look at homology, you can see the different forelimbs of these different organisms. We're comparing the human with the mouse with the bat, and there's definitely some similarities in terms of the bones that are present between the three. But the analogy is going to be more based on where these organisms inhabit. In other words, the type of environment that they're in. So when you look at a bat wing, you make a comparison to a butterfly wing, and you make a comparison to a bird wing. The environment that they live in all tend to encourage the idea of flight. But the way that these organisms actually fly are very different, and the structures that they use are very different as well. So this would be considered an analogy, in other words, a comparison, just simply due to convergent evolution as opposed to homology. And as we had said, convergent evolution is going to be simply similar environmental selecting agents that might sort of influence the characteristics that we see. So convergent evolution is going to occur when similar environmental pressures, in other words, natural selection, are going to produce similar analogous adaptations in organisms from different evolutionary lineages. And in the previous screen, we had looked at the comparison between the bat and the bird wings. These are considered homologous as forelimbs, but they're analogous in terms of the function that they provide. Now, analogous structures or molecular sequences that evolved independently, sometimes we give them a special word and we call them homoplasties. Now, homology can be distinguished from analogy by simply comparing fossil evidence, and we can look at the degree of complexity between those different pieces of evidence. Basically, the more complex two similar structures are, the more likely it is that they are homologous to each other. Now remember, there is an area of science called molecular systematics, and this is going to use DNA and other molecular data to help us to determine evolutionary relationships between these organisms. Now, once homologous characters have been identified, those can now be used to infer a phylogeny and help us create those phylogenetic trees. So the next thing we're going to look at is something called cladistics. Now, cladistics is just simply a method that is involved when you look at the systematic relationship between organisms. And so cladistics is going to be used to group organisms by common descent, which is something we've already talked about. Now, when you talk about cladistics, we often talk about something called a clade. Now, a clade is going to be a group of species that includes the ancestral species as well as all of its descendants. Now, clades themselves can be nested in larger clades, but not all groupings of organisms are going to qualify as a clade. So down here towards the bottom, we have three different types of groupings that you're looking at. One of those is monophyletic, the second is paraphyletic, and polyphyletic is going to be the third. Now, when you look at the prefixes, mono is going to refer to one, para is going to refer to besides, actually, and poly is going to refer to many. Now, a monophyletic grouping is going to signify that it actually consists of the ancestor species and all of its descendants. And so if you look at this very first grouping, the ancestor species is going to be located right here, and this ancestor species is going to be related to both A, B, and C. Now, when you look at paraphyletic grouping, this is going to consist of an ancestral species and then only some of its descendants. And so if you look here, we leave G out and we only include comparisons between D, E, and F for that particular grouping. But over here on the right, if you look at a polyphyletic type of grouping, this is going to consist of various species, but they actually lack a common ancestor. And so the big thing to recognize on this one is if you look at C, C does not necessarily have a common ancestor recently with D, E, F, and G. And so when you look at that type of comparison, that's considered a polyphyletic group. Now, as you go and you create these different phylogenetic trees or cladograms, what we're going to do is, again, we're always going to compare the different characters or characteristics between those different organisms. So in comparison with the ancestor that we're looking at, an organism has two things. It has both shared and different characteristics. Now, a shared ancestral character is going to be considered a character that actually originates in the ancestor of a taxon. Now, in comparison, a shared derived character is going to be one that is rather unique to that particular clade. Now, remember, a clade is going to be basically organisms that are within the same grouping with that common ancestor. Now, down here towards the bottom, this is going to be a diagram that's going to represent sort of what we do when we try to make comparisons between those different characters or characteristics. And the very first thing that we'll often do is we'll put it into a table. So this is going to be considered a character table. Now, the zeros that you see here are going to represent basically the characters that you see on the left and whether or not the organisms on the top have that character. So if you see a 0, it means they don't have the character. If you see a 1, it means they do have the character. So if you look at the lancelet, you're going to notice that it has all zeros, which means all of the characters that we're looking at, the lancelet does not have any of them. But if you look at the lamprey, you're going to notice if you make comparisons between the lamprey and the lancelet, it actually does have a backbone. If you look at the tuna and you make comparisons between the tuna and these two previous organisms, it actually has a backbone, but it also has hinged jaws, which is something the lamprey doesn't. And so what we do is we take this information and we create our phylogenetic tree. And so we take the characters on the left and we put those on the left of our phylogenetic tree. Depends on how the tree is set up. And of course on the right we're going to have our different organisms. And so if you notice, this is going to be our ancient common ancestor for all of our different organisms. The first character is going to be that backbone, and if you notice, the lamprey, the tuna, the salamander, the turtle, and the leopard all have backbones, but not the lancelet. And then we're going to look at the next character, which is the hinged jaws. And as we had said from our table, lamprey does not have hinged jaws, but the other four organisms do. Then we continue down and we look at the walking legs, and of course the tuna doesn't have walking legs, but the salamander, the turtle, and the leopard do. We look at the amniotic egg, which means that the turtle, the leopard do have amniotic eggs, but the salamander does not. And then again we look at that shared derived character, which in this case for this leopard is a really unique character because it's the only one in our cladogram or phylogenetic tree that actually contains hair. And so that would be the method that we would use to actually create the cladogram that you see, or the phylogenetic tree that you see over here on the right. So when we do get into the idea of cladistics or creating those phylogenetic trees or cladograms, what we need to think about here is the idea of maximum parsimony or maximum likelihood. And so what this is going to imply is it's going to make the assumption that the tree that we do create is going to be the tree that actually requires the fewest evolutionary events. In other words, the appearances of those shared derived characters that we had looked at in the previous screen. And so down here towards the bottom, again, we have a good example of maximum parsimony or maximum likelihood. And one of the ways that we actually make this a little bit easier is now, because of the technology that we have, we can make comparisons between the particular DNA sequences between different organisms. And so the creation of these particular cladograms or phylogenetic trees actually becomes much, much easier. And so if you notice, based on the table that we have here, if you make comparisons between the human and the mushroom, you're going to find there's about a 30% difference in the DNA between these two organisms. If you make comparisons between the human and the tulip, there's actually about a 40% difference between those two organisms. And what we do is we take that information and we look at the possible trees that we could create from that information. And so if you notice, we have one that's more likely and one that's less likely. And so as we create our tree, if we create it this way, then we would put the fungus or the mushroom right here, and then we would put both of these organisms in the same clade. So we'd put humans and tulips in the same clade. So if we look at that, what that's going to indicate to us is if we have both of these within the same clade, that sort of implies that they are more closely related. Well, if you look at the comparison between the human and the tulip, it was about 40% in terms of differences. But if we sort of rearrange this and we put the human with the fungus or the mushroom, there was really only a 30% difference between those two. So it's much, much better to put the cladogram or the phylogenetic tree in this particular type of way as opposed to this way, because this one definitely makes more sense in regards to those DNA sequences that we would look at when making those comparisons in their DNA. Now, another thing that we look at is something called phylogenetic bracketing. And what this allows us to do is after we create our phylogenetic tree, we can actually make some predictions of the features of that particular ancestor for those organisms. And so if this is considered the common ancestor for all of these organisms you see here, looking at the characters or characteristics of these organisms, we can sort of infer possibly what that ancient common ancestor might have looked like. So that's considered phylogenetic bracketing. Now, as we go ahead and document the evolutionary history of an organism, what we're going to find is that we can actually now make comparisons, as we had said, of those nucleic acids. And as we make those comparisons, we're primarily looking at the DNA, but we know that that DNA is going to code for various forms of RNA. Now, when we look at ribosomal RNA, for example, we're going to notice that that particular RNA does tend to change relatively slowly. And what we can do is we can actually use this information to investigate the branching points hundreds of millions of years ago because of that incredibly slow change. But then we have a type of DNA called mitochondrial DNA that actually evolves much, much more rapidly. And with that type of DNA, we can look at the most recent evolutionary events or history of an organism. Now, something else that we can look at when it comes down to making inferences about the evolutionary history of an organism is we can look at something called gene duplication. Now, when we look at gene duplication, basically what we're thinking about here is that the increase in the number of genes in that genome could possibly provide more opportunities for that evolutionary change. Now, there's two different categories of genes. One of those is called orthologous, I think, genes, and the second is called paralogous genes. Now, when you look at these particular types of genes, they're going to be found in a single copy in that genome of that organism. And when you make comparisons between groups of organisms, they do tend to be homologous between those species. In other words, they're very similar. But if speciation does occur, then they can actually diverge from each other and maybe function in a little bit of a different way. The second grouping is going to result from something called gene duplication. And so these are going to be found in actually more than one copy in the genome of that organism. So what they can do is they can actually diverge with that particular clade or grouping that carries them, and they can often evolve into new functions within that particular grouping or that species. So this particular diagram is simply going to illustrate the difference between these two different types of genes. And if you notice, both of these particular types of genes are going to originate from the same ancestral gene or ancestral species. And if speciation occurs, what you're going to find is there will be a divergence of that gene. And if we make comparisons between species A and species B, you're going to notice that the colors are a little bit different, which basically means that they're homologous to each other, but maybe the function of that gene is a little bit different from each other. But when you look at these particular types of genes, we're going to look at species A here. You're going to notice that that particular gene has now been duplicated. And when it was duplicated, it actually diverged within the same organism. And so now instead of having one copy, we actually have two. And if you notice, the color's a little bit different, which indicates to us that the function now of that duplicated gene might also be a little bit different as well. So now thinking about all of the information that we've collected up to this point, what we can do is we can take that information, and some scientists have actually created something called a molecular clock. Because what they've noticed is that some genes appear to evolve, it seems, at very constant rates. Now they can use this information to sort of estimate the dates of past evolutionary events, and that's what you're seeing down here towards the bottom left of the screen. Now, because of this, they have created something called the neutral theory. Now, the neutral theory is going to state that much of the evolutionary change in genes and proteins has actually no effect on fitness, and therefore is not going to be influenced by Darwinian natural selection. Now, what this does is it essentially states that the rate of molecular change in these particular genes and these proteins that are created by these genes should be regular like a clock. And as I had said, the HIV virus down here towards the left is a really good example of that. So what scientists have done is they've actually looked at the information that they've collected in regards to changes in this HIV virus between the years of 1980 and in this particular graph around 2000. And if you notice, we have lots of changes that have occurred or been clustered around this area. So as we had said, we can take this molecular clock and we can estimate the dates of past evolutionary events. So the idea here is that we sort of extrapolate and we bring this information in a linear fashion all the way back, and around the 1930s possibly, this would be the first indicator that the HIV virus has actually came to be. Then of course over many years the change has occurred. But this would be the first period of time that we've actually had evidence in regards to data collection on this particular virus. Now over here on the right, this is going to be another good example of a molecular clock. So we're looking at the change in different mammal genes over millions of years. And again, looking at these mutations that have occurred over this period of time, you're going to notice it does appear to be in a rather linear fashion. But remember, natural selection is out there, so it does have some influence on this clock. And so if you notice, down here towards the bottom, we have 3 genes that are in green. This is going to indicate that they don't necessarily follow the path that you see for the other changes that have occurred over this timeline. And so we would make the assumption that they have evolved a little bit slower, and because of that, in this particular case, the characteristics that maybe have been coded for by those genes, it has caused this particular group to diverge from the timeline that you see indicated on this graph. Now the very last thing is just a really quick look at the domain system that you would find in regards to the different organisms that you find on this planet. Because there have been some very substantial interchanges of genes between organisms, we have these different domains that have been created. And some researchers have actually suggested that the eukaryotes, in other words, what we are, and all of the what we would consider more complex organisms actually arose as a result of a process called endosymbiosis. And that endosymbiosis actually occurred between a bacterium and what they would consider archaeum. Now the 3 different systems that are accepted now by scientists are eukarya, the archaea, and the bacteria, which you see down here towards the right. And if you notice, all 3 of these have that ancient common ancestor to all of life. Now, as we had said, endosymbiosis is what some scientists believe, actually a lot of scientists believe, that actually created the eukaryotes, which are going to be, again, this particular domain. So endosymbiosis basically means within, and what that indicates to us is that we had members from this particular group and members of this particular group that actually came together. And because of that sort of combination between those different members of those domains, they created what you see up here towards the top of this particular diagram. Right, so that's going to finish up our screencast for Chapter 26, and as always, please make sure that you have completed your screencast study guide.